Hello, my dear students. This time we're going to talk about a new topic, which is the chromosomes and gene expression. I know these terms are so familiar with you, but I'm hoping that this discussion will give you additional information and knowledge about the said topic. So, by the way, I am Javelin A. Tisado, a third-year Best Gen Sci student, your reporter for this time. To understand what is this all about, let's take a sort of review about chromosomes. So, what is chromosomes, by the way? Well, basically, chromosomes is a thread-like structures that is usually found inside the nucleus of an animal's and the plant cells. As you can see here in the picture, this is the cell. Inside the cell, there is a nucleus in it where chromosomes are actually found. However, in this picture of a chromosomes, we can see its different parts. So this is the P arm, the Q arm, the centromere, where it links the sister chromatids together. So this is the chromatid and the other chromatid. That is why we call it the sister chromatids. Furthermore, one of the functions of chromosomes is to keep the DNA tightly wrapped around the spool-like proteins called the histones. So, if we're going to zoom in the structures of the chromosomes, we can see the proteins, which is the histones. So, this is the nucleosome also. It contains the number of histones in which it makes a subunit of the chromatin. So, this is the whole structure. We call it the chromatin. Take note that each human contains 46 pairs of chromosomes in total, where the 22 pairs of it are called autosomes and the 23rd pair are it is basically called the sex chromosomes. Now, what is this gene expression? Well, it is a process by which our instruction in our DNA are converted into functional products such as protein. In general, DNA is replicated to make mRNA, and this mRNA will be converted into proteins. Remember that proteins are large, complex molecules that play many critical roles in our body. They do most of the work in cells and are required for the structure, function, and even for the regulation of the body's tissues and organs. However, this process is very important because this allows the cell to respond to its changing environment. It acts as both an on and off switch to control the number and volume of the protein synthesis also. In other words, gene expression is tightly regulated. And lastly, it involves two key processes such as the transcription and translation. Remember that transcription comes first before translation, as C comes first before L in the alphabet. Now let's proceed to transcription, where the DNA in a gene is copied to produce mRNA through an enzyme RNA polymerase. When the RNA polymerase found the region that signal to start making the replication of the DNA to produce mRNA, otherwise known as the promoter, the polymerase will bind to it and would incredibly start the process of transcription. The polymerase would take the chance to produce mRNA, and so mRNA will bring the transcribed message to the ribosomes, because in this case, whole DNA can make it to go out wholly from the nucleus, but through mRNA, it would be possible for it to travel from the nucleus of the cell out to the ribosomes for the preparation for the next process of protein synthesis, which is the translation. Right after the mRNA bring the transcribed message to the ribosomes, where protein synthesis usually occur, translation starts right away. And so the message carried by the mRNA is read by a carrier molecule called transfer RNA or tRNA. However, mRNA is read 3 letters which commonly called a codon, and each codon specifies a particular amino acid. When the mRNA sequence is read, each tRNA molecule delivers its amino acid to the ribosomes and binds temporarily to the corresponding codon on the mRNA molecule. Remember that amino acids is the building blocks of protein, and so this amino acid will join together into a long chain called a polypeptide, which eventually the proteins itself. As what I have mentioned earlier, that gene expression is tightly regulated. This is commonly called as gene regulation. And what is this gene regulation, by the way? So it is the process used to control the timing, location, and the amount in which gene is expressed. The process can be complicated and is carried out by a variety of mechanisms, including through regulatory proteins and chemical modifications of DNA. Gene regulations is the key to the ability of an organism to respond to its environmental changes. To fully understand what really happened during the regulation of the gene expression, let's take time to understand this video instead.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase, and poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
However, these processes quietly occur inside our body. So this protein synthesis or gene expression are often called as the central dogma of molecular life. For more learnings, I have here a supplemental video entitled The Central Dogma of Life. And lastly, these are my references to make this presentation possible.